Welcome students to today's session. We shall start with a new chapter in biology, tissues. In your previous lesson, we have discussed about the cell components, types of organism based on the number of cells, unicellular, multicellular. But in multicellular organisms, millions of cells, and most of these cells carry out many each specialized function is taken by a group of cells. These group of cells carry out this function very efficiently. Like for example, in humans, cells are involved in contraction to cause movement. The nerve cell carries out messages. And in plants, the vascular tissues, xylem contains water and minerals. Phloem contains prepared food materials. This means that a particular function is carried out by a cluster of cells, which is located in a definite place in the body. This cluster of cells are called as tissues. They are arranged and designed in such a way that it gives highest possible efficiency of function. Hence, a group of cells that are similar in structure and work together to achieve a particular function forms a tissue. This lesson, we are going to study the different types of tissues occurring in plants and animals. Composition of these tissues. Tissues are a group of cells having similar structure and similar function. In plants and animals, group of cells called tissues carry out specific functions. I have given you some examples over here: the nervous tissue, the muscular tissue, and the meristematic tissue. You have got the muscular tissues picture over here and the nervous tissue below that. You can look, they are a group of cells clustered together in a particular place and they do a specific function. They cannot deviate and carry out other functions. Like muscular tissue is for contraction and relaxation. It is designed in that way. You can see the spindle shaped cells and it is designed in that way to do the function of contraction and relaxation. The cells of nerve looks like wires and cables and they are organized in such a way that they are involved in transmission of messages. So they are designed for a particular function and they perform this function in groups known as tissues. Let us now look at the classification of plant tissues. Plant tissues are mainly classified into two categories: the meristematic tissues and the permanent tissues. The meristematic tissues are undifferentiated cells. That means they can differentiate and grow into different type of cells. So the meristematic are the first born cells and they are undifferentiated soft cells, and you have got three different types of meristematic tissues, that is apical meristem, intercalary meristem, and lateral meristem. These are the three types of meristematic tissues: apical meristem, intercalary meristem, and lateral meristem. Now we shall go into the next category, permanent tissues. Permanent tissues are differentiated cells, not like meristematic cells. They are differentiated and organized to perform certain functions. So they are of two types main: simple permanent tissue and complex permanent tissue. So you have got simple permanent tissue and complex permanent tissue. Simple permanent tissue is further divided as parenchyma, collenchyma, and sclerenchyma. Complex permanent tissue is divided as xylem and phloem. We shall look into details of these different types of tissues and its classification. First, let us study about the meristematic tissue. Meristematic tissues are found in the growing regions of the plant, like tips of fruit, stem, and branches. So I have told you they are undifferentiated tissue, very soft tissue, very much rich in protoplasm. They divide continuously to help the growth of the plant. Now let us see where they are located in the plant. The apical meristem is located in the root tip and the shoot tip. Okay, so they are involved in growth of the plant both upward and downward. They are present in the root tip and the shoot tip. The next one, lateral meristem, is present in the side of the stem. Lateral meristem is present in the side of the stem. They help to increase the grit of the plant. The intercalary meristem is present at the base of the leaf. This is intercalary meristem. It is present at the base of the leaf and internode. This is internode. The place where the leaf starts to come out is called as the internode. It helps in the growth of these parts. So together all these three meristems help in growth of the plant both lengthwise and breadthwise. So now you have studied the three different types of meristematic tissues: apical meristem, lateral meristem, and intercalary meristem. The numbers are not to be read. Okay, shall go on to the next type, the permanent tissues. The permanent tissues are formed from meristematic tissue, that is, when they differentiate, they grow up and differentiate, they form permanent tissues. They do not divide and have permanent shape and size. They differentiate into different types of permanent tissues. Permanent tissues are of mainly two types. They are simple permanent and complex permanent. So simple permanent is made up of three types of cells: the parenchyma, the collenchyma, and the sclerenchyma. We shall study in detail about these three different types of cells. First, let us look at parenchyma. Parenchyma consists of loosely packed thin wall cells having more intercellular space. Some parenchyma cells in leaves contain chloroplast and prepares food by photosynthesis. Some parenchyma cells have large air cavities which help the plant to float on water, and they are called as aerenchyma. Some parenchyma cells of root and stem store water and minerals. So this is the structure and function of parenchyma. Now let us see the next type of cells, the collenchyma. These consist of elongated cells having less intercellular space and are thicker at the corners. They are flexible and allow easy bending of different parts of stems and leaf. They also give mechanical support to the plant. The next category is sclerenchyma, consists of long, narrow, dead, thick-walled cells. They are usually found in fibers. The cell wall contains lignin, a chemical substance which acts like cement and hardens them. It gives strength and hardness to the plant parts. So these are the three different types of cells. These are all you will study in today's session. The next slide I will show you how to draw these structures in your notebook. Look at parenchyma. It is to be drawn with this format. You can see the cells. The cells should have only three cells meeting at a point. Can you see it forms a triangle between the three cells? You look at any such intersection, it should have only three cells joining at. See, there are only three cells meeting at this point. This is how you draw it. If you start drawing it like this, one after the other, then you will not get parenchyma. You will get stacked idlis. You should not draw it like this. It should be drawn only 3 cells meeting. If you draw it like this, then there will be 4 cells meeting at the point. Can you see there is a 4 cell meeting point here? It shouldn't be drawn like this. It should be drawn with the cells having, that is 3 cells are joined at a place. This is how it is to be drawn. Be very careful while you are drawing the structure of parenchyma. It is dense with cytoplasm, with a nucleus, and there is a lot of intercellular space as in the form of this triangle. These are the intercellular spaces. Please draw it properly in your notebook. All diagrams should be very neat, then only it gives you an impressive work. Now this is what you see on the top is a cross section, that is when you cut it cross, and what you see down is the longitudinal section, that is when you see it in sideways. So if you want to see a cross section of this, then it is cut like this. This is how it is cut. So instead, this top one is a cross section, this second one, down one of parenchyma, it is a longitudinal section. So please be alert in doing the diagrams. Look at the next one, collenchyma. You have to make the end walls thicker. This is lignin deposit. You can make the end walls thicker. It has got a structure like parenchyma, but the end wall thickenings are there. End walls you can make it thicker. All the end walls can be made thicker. That's the difference between a parenchyma and collenchyma, because it gives mechanical support. It is having this sort of a structure. Sclerenchyma is total fibers. It is not living cell. It's all dead cell with rich deposit of lignin. The entire wall is thickened. That brown structure is fully, the brown colored structure is fully thickened end wall. They are fibers, sclerites, all these are having this sort of structure. No protoplasm, nucleus or cell contents inside. It will be just empty space. Fibers. Okay. And these are stone cells that you see here. They are also sclerenchyma cells. This picture gives you sclerites, fibers and stone cells picture. You need to draw only fibers. Please draw it as it is given here. The lumen for stone cells are very less. Lumen means the empty space between is very, very less. For fiber it is maximum. For sclerites it is little more less. That means there is thickening of wall more. And for stone cells it is completely thickened. These stone cells are found sometime in certain banana fruit where you will find thickened fibers. Okay, they are these stone cells. So this is how you draw the pictures in your class workbook. Thank you for attending today's session. Hope you have enjoyed the session. Thank you once more.